Austin, start us off.
They're surrounded by membranes.
Okay, you're telling me what they have, but I'm asking what they are.
They make living things. They are like what living things are composed of.
Okay, good. That's a good start there. Charlie?
Microscopic organisms.
They're are they all organisms? Every cell?
No.
No. Okay, Ty?
No.
Sam?
They have nucleus.
That's what they're made of. Again, you were closer on your first one there. Lindy?
Cells are what make up living things.
Okay. So kind of what Sam said there. So let's start with this. We're going to define a cell as a basic unit of structure and function. Now, anytime I have defined a cell as a basic unit of structure and function, the thing that I find students struggle with is what do we mean by structure and what do we mean by function? So the next thing I want you to do is I want you to define at your tables what is structure, what is function. Go. Chase, what do we mean by structure?
How it's built?
Okay, so how it's built. What else do we mean by structure? Sam?
It's like what a certain family, it's a certain asset of a certain family, what they share. Like everything in it has that specific thing.
You're not telling me what structure is. You're just saying things that have it. Okay? Ty?
Like a building.
Okay. Whitney?
Is it like a shape?
Okay, like the shape. There you go. That's closer. Lindy?
Structure is like the skeleton of a body or something. It's like what makes up the shape, and it's what scientists look at when they do like similarities to pictures. They look at like.
Okay. So when we talk about a structure, we're talking about what it's made of. And one other thing here, very important, and how it's put together.
TV just.
It'll come in and out. You'll just have to kind of hang with me. Function is what? Sam?
It's like how something works.
Okay. How it works? Kenny?
How it like moves and how it like responds and how it adapts.
Okay. David?
Probably it moves and processes.
Okay. Lindy?
It's how it like works, like the function of like. I don't know how to explain.
Okay. So when we talk about function, we're talking about the process or activity, if activity is an easier word for you, which enable the cell to grow and reproduce. What we're going to talk about is the cell theory. Okay, so basically the cell theory was devised after years and years and years of research, and scientists discovered that cells have basic things about them. So the first thing is that all living things are composed of cells. So everything that's living is either made of one or more cells. They're either unicellular or they're multicellular. Whoops. The next part of the cell theory is that cells are the basic units of structure and function for living things. Okay, so all the way down to a single cell or a multicellular organism. It's the basic unit of structure and function for living things. And then the last one, which is pretty intuitive, but it kind of reminds me of when we talked about where life comes from. We said all living things come from other living things. It doesn't just come out of nowhere. Cells are produced from other cells. That we're going to talk about or focus on is how do these microscopes work? And we're going to break this up into two types of microscopes. The first one is going to be like the microscope we use here in this classroom, and it's called a compound microscope. And the second one is what we call a scanning electron microscope. And essentially this compound microscope works by just sending light through a lens. And when that happens, you're looking at a very thin specimen and you're able to view it at a magnified level. So to magnify and view it. Scanning electron microscopes, on the other hand, they send little beams of electrons at an object and they bounce back to give an image. So we'll kind of take a look at what that means. But this is operating under beams of electrons. As a reminder, an electron is a really small particle. It's part of an atom. You have the nucleus in the middle holding the neutrons and protons. Then you have the electrons and speed and motion around the outside. And they are a particle, meaning they are made of matter. They take up space. They have a mass to them. Okay, so some of the images in which you can get from them. If we take a look at this example, this is an example of a microscope from a compound microscope. And when you look at this, what is it that you think you're seeing? Talk at your tables. What do you think you see up here? So what you should get from this when you're looking at it, you're going to be able to see that obviously these are all individual cells and they're connected, right? But this is actually like a cross section where you would cut like a piece of plant tissue really, really thin. And you're seeing in the middle here, these larger sections would be what's called a xylem, and these smaller cells around the outside are a phloem. Not that you need to know that, but what scientists struggled with when they first started viewing these things was what the heck is it? So they would look at different things and they wouldn't know exactly what they were looking at, and it took them a lot of years to figure out exactly what they were looking at. Because of that, they started developing more advanced technologies, more intricate ways to look at things, and that's when the scanning electron microscope came about. And that was a way to look at things almost in a three-dimensional object, things that were maybe too small to see with our own eye, but we wanted to see them through three-dimensionally. This would be an example of an image that they would receive back from a scanning electron microscope. So talk at your tables. What do you think this is? When you're looking at this, actually what you are seeing is a fly, and more particularly is a way for them to understand how the eye works. So this whole thing right here is an eye. And as you can see, it's made up of all these different lenses that come together to make a compound eye. And before this, scientists weren't able to really determine how flies saw things or how they could see things and how they could see things essentially behind them. Because if you look at the shape of that eye, it's cupped, right? And they have lenses allowing them to see out in different angles. So we can see roughly 180 degrees, right? Your hands out to the side, you can probably see those. But if you take them back behind the sides of your eyes, you really can't see it if you're looking straight out. A fly would be able to see this, okay, without turning its head. And that's some of the different things that would be some of the different things that scientists would be able to see once they were using these higher technologies or more complex technologies. And at the bottom of your sheet, you guys see where it says calculating the power for following objective lenses, or for the following objective lenses. And what you guys have on the microscope is two main lenses. The one up towards your eye is called the what?
The Ocular lens.
Yeah, the ocular lens. So we have ocular lens. And then you're kind of looking through that, comes down, I don't know. You guys know how terrible I am at drawing, right? And you have these
How if you
three.
looked at the microscope? It's right there.
I don't need to look at it. You have these three ones down here. What are these ones called? Lindy?
Objective.
Objective lens.
Your objective lens.
I realize.
Just remember, your objective lens is nearest the object. So it's the lens by the object, the objective lens. Now, each lens is going to magnify things. Most ocular lenses are going to magnify things 10 times. For your objective lens, it just depends on the lens. You might have one lens that does it 4 times. One lens might just do 10 times larger. And maybe the third lens does 40. So one of the things you guys need to know is, well, how much larger is the object or smaller is the object than what I'm viewing it? And in order to determine that, you're going to have to do some math. So how we break this up is we look at, okay, our objective lens, or excuse me, ocular lens is 10 times larger, meaning I have one sheet of paper, and I look at that sheet of paper just through my ocular lens. It's 10 pieces of this piece of paper larger than what it normally is, right? And then we have that times whatever objective lens you're using, okay? So if I was using a 4 times objective lens and my ocular is already multiplying it 10 times, what's my total magnification? Sam?
40.
Yeah, it's 40. You multiply it. So you have 10 times 4 equals 40 times larger. Okay? So what you need to do now is figure it out. For your other 2 objective lenses, the 10 power and the 40 power, what's your total magnification? Go ahead and do that on your sheet right now. Again, you're taking your objective lens times your ocular lens. So if your objective lens is 10 times, you're going to take your 10 times for your objective lens times the ocular lens, which was 10 times. And that's going to be what? 10 times 10 is? 100. Okay, so then we're going to do the next one, the one that's 40 times. So we have 40 times 10. And that's going to be 400. Good. So that's how you would calculate the magnification of an object that you're looking at and determine how big or small it really is. So then the other thing we need to talk about is these different terms. We already said that this top lens is the ocular, and these guys down here are the objective lenses. Some other terms that we use right here in the middle portion, we actually call this the body tube. This piece right here that rotates, hint, hint, rotates, is the revolving nose piece. This piece over here that's the larger of the 2 focuses, large focus is called your coarse focus. And this little guy down here is actually going to be your fine focus. We carry the microscope holding on to what we call the arm. And we place our hand underneath the base. The piece down here at the bottom, very simple, it's our light. And then the black piece in which we set our specimen on is called the stage. And then on our stage here, you can see these silver little clips. Those are our stage clips. And of course, we can't talk about the microscope without talking about two really famous names. The first guy is Robert Hooke. He designed the microscope. I'll say designed / built because he actually made one. But he really didn't know what he was looking at. He would look at different things underneath the microscope, and he might know where it came from, but he didn't necessarily know what all the different things was that he was seeing. It's kind of like when we looked at that image of the plant cross-section, you guys were like, well, it's cells, but I don't know where it's from, or I don't know what exactly it is. Until Antoine van Leeuwenhoek came along, we didn't really know a lot about the microscopic world. And that's when he first identified cells as living things. So he found some of the first unicellular organisms and was able to identify what a cell was. Now, I joke and I say we just crammed like 30 years of science history into all of like maybe 2 minutes and explaining those two people. So there's a lot more to the story, a lot more information out there. But the big picture is that we first had to have the tool. So we had Robert Hooke, who was really, really important for that. And then after he provided us with the tool, we had other scientists who started really investigating things at the microscopic level. And it's a huge field of science today. So it's something that's really, really important for that.